Welcome to Inside CRISPR, a found object experience. Karate chop the floating DNA helixes to begin. Throughout this experience, when you see this icon, you will need to pinch your fingers to advance. Let's try it. By the way, these little orbs are pulsars. They use concentrated gravity to pull light particles out of a strong light source. They rip and bend the light to create these visuals. In 2020, Dr. Jennifer Doudna and her colleague, Dr. Emmanuel Charpentier, won the Nobel Prize in Chemistry for their discovery of the CRISPR-Cas9 gene editing technology. News of the discovery shook the scientific community. But what is CRISPR-Cas9 gene editing? How does it work? And what potential does it hold? What follows is an interactive summary. The full experience lasts nearly 10 minutes. To understand CRISPR-Cas9 gene editing, we must review how nature already performs gene edits with no human intervention. Let me repeat that. Nature already performs gene cutting and splicing. To see how, we must zoom in on the great, invisible war between bacteria cells and the viruses that attack them. Use your hands as paddles. See if you can deflect one of the floating balls to ring the bell. Every moment of every day, there's a battle raging between viruses and bacteria cells. Scientists estimate that as many as 40% of bacteria cells in the ocean die every day under attack from viruses. These viruses that attack bacteria are called bacteriophages. Bacteriophages have evolved to hijack the bacteria cell and use its machinery to reproduce their own viral DNA. Before long, the newly manufactured bacteriophages will burst out, destroying the old cell, and start the cycle again. Reach out to poke this bulging bacteria cell. So, we've learned that there's a never-ending battle between bacteria cells and viruses called bacteriophages. But what does any of this have to do with gene editing? Let's find out. The viruses are so brutally effective at replicating their DNA, it is a wonder that the cells stand any chance at all. But they do. The bacteria cell has a sophisticated defense against the virus. First, it collects small snippets of viral DNA and keeps them on record. This way, it knows how to recognize viral DNA. Drag and drop each DNA strand into the corner of the cell. Good! Each snippet of DNA is a unique signature for a type of invasive virus. This collection of viral DNA snippets, held in the corner of the cell, is called CRISPR, clustered, regularly interspaced, short palindromic repeats. Each snippet is a unique identifier for a particular type of invasive virus. Think of CRISPR as a collection of wanted posters in a police station. Now make your fingers into a gun. Shoot only the viruses shown in the wanted posters by bending your thumb. Do it before they can hijack the cell. You've taken the first step. You've identified viral DNA using the cell's CRISPR as a reference. But let's take a step back. How does the cell find and isolate those short snippets that end up in the CRISPR sequence? To generate a CRISPR strand, a cell must be able to do the following. 1. Identify a viral signature. And 2. Cut it out. Luckily, the cell has Cas9 proteins. Cas9 proteins are extraordinary. These proteins move quickly over a DNA strand to identify viral sequences. Even more astonishing, they can act like scissors to cut out sections of DNA to add to the CRISPR reference. Let's see it in action. You're going to help the Cas9 do its job. First, use your pointer finger to draw a DNA strand. Wonderful! Pinch again to move. The Cas9 can precisely cut out viral DNA sequences to add to the CRISPR collection. This natural process caught the attention of Doudna and Carpentier. Let's do it again. This time we'll help the Cas9 cut out the viral signature. First, use your pointer finger to draw a DNA strand. I've started the Cas9 moving slowly. When it gets to the beginning of the viral sequence of DNA, I want you to pinch quickly, like this, to make the first cut, and pinch again to make the second cut. Pinch again to move to the next section. Do you see how it's cutting out the bad sequence of letters to add to the CRISPR strand? Luckily, Cas9, the genius little protein that it is, can also replace or repair the sections that it cuts out. But how? Let's think about this. In order for the Cas9 to know how to repair the DNA, it needs a set of instructions. Let's do it again, this time giving the Cas9 specific instructions. Use your pointer finger to draw a DNA strand. This time, we're going to replace the missing section of DNA with a more desirable. Once the Cas9 is moving, you will pinch and hold down to repair the broken track of DNA. Pinch again to move to the next section. Good, you're doing it. Doudna and her lab mates were quite stunned by this realization. They had set out to study how bacteria defend themselves against bacteriophages. Along the way, they found a crack in the fabric of creation. It's worth noting that just because we know how to cut out and replace sections of DNA does not mean that gene editing is easy. Far from it. There are over 100 million DNA letters in each person's unique DNA, spread out over 23 chromosomes. Sometimes it's relatively simple to know which DNA letters contribute to individual traits. But more often, many different DNA letters inform even a single trait, like eye color. It will take scientists a long time to figure out how these hundreds of millions of individual letters in our DNA affect the traits that appear in our bodies. There are several known genetic diseases that have a very direct genetic fix. For example, sickle cell anemia and cystic fibrosis are single gene defects. Genetic modifications to mosquitoes may not be a genetic cure, but it is an effective way to curb a disease. Scientists can modify mosquitoes using CRISPR so that they are 99.5% less likely to carry malaria. If mosquitoes pass the trait down through generations, soon no mosquitoes will carry malaria. In agriculture, CRISPR can be used to modify crops to be more resistant to disease and infection. A CRISPR-Cas9 modification to barley confers a resistance to a powdery mildew that ruins the barley crop. This means that a single harvest can feed 15% more people. Here is a more controversial but still widely discussed concept. Animals such as pigs can be genetically modified to carry organs that can be transplanted into human bodies. These xenotransplants are on the rise and could eliminate wait times for people in need of vital organs. The potential is breathtaking, as are the dangers. CRISPR is a surprisingly accessible technology. Even today, amateur scientists and tinkerers can make their own experiments. In the wrong hands, this could be catastrophic. We don't know how this will play out, but we do know that we can never put the genie back in the bottle. The discovery of CRISPR gene editing will alter the fate of life on our planet. Thank you for playing.